Hello. In our case study today, we're going to be talking about fluorouracil. This is a drug that is used to treat cancer patients, so specifically patients who are diagnosed with colon cancer, rectal, breast cancer, pancreatic cancer, all of the things that are sort of happening in the gut area. So we're going to talk a little bit about how this drug is metabolized by cells in order to make an inhibitor. We're going to talk about how fluorouracil inhibits RNA synthesis and the mechanism for how this inhibition occurs. So fluorouracil is inactive. It's an inactive substance until it actually is metabolized by the human body. And then it produces the active form of the drug. It is a nucleobase analog that is uracil, so it sort of takes the place of uracil. And so sort of the mechanism of that, or sort of the structure of that, is the hydrogen at position 5 is replaced by fluorine. Once it's in the body, fluorouracil is converted into an active enzyme, or an active metabolite, and that is called FUMP. And another word to talk about fluorouracil is just saying 5-FU, since it's sort of a long term. The target cell is what converts the 5-FU into its active form. So when we're looking at this photo here, we can see how we have the 5-FU. This is sort of the molecule, and then once it crosses into the target cell, it is converted then into the 5-UMP. And then what it ends up doing then is, once it is actually inside of the body, it converts into the active deoxynucleotide, and it inhibits DNA synthesis. So what it's doing there to inhibit DNA synthesis and RNA synthesis is it blocks the conversion of deoxyuridylic acid into thymidyllic acid by the cellular enzyme thymidylate synthase. So we're going to see a sort of a structure of that and what that mechanism looks like. So sort of how it inhibits and the mechanism of that sort of correlate. So by blocking DNA synthesis and RNA synthesis, it is going to slow tumor growth. So once the synthesis is unable to carry out, the tumor growth doesn't happen as rapidly as we see with cancer cells. So what exactly is happening here? What is the mechanism? So once it's in the body, it is converted into that active metabolite, which we talked about, which was that that FUMP. So here's sort of what we're seeing here. It's converted there. It ends up replacing uracil, right? And then it's incorporated into RNA. So this sort of a fake for uracil when it's being placed into RNA synthesis, and it's being matched during transcription. So it ends up replacing itself in there in the place of uracil. It's being incorporated into the RNA, and it inhibits RNA processing, which then we can't have any cell tumor growth. There's another active metabolite that we're going to see on this chart here, that FDUMP. That's another one that is created that once it's inside of the body, it inhibits that thymidylate synthase, resulting in deletion of TTP, which is one of the 4 nucleotide triphosphates used for the synthesis of DNA. So not only are we getting in the middle of RNA synthesis, we're also damaging DNA synthesis. So it's sort of twofold what's happening here. So we end up having this increased expression of thymidine synthase, and then it ends up causing DNA damage. And just the mechanism of having the sort of fake uracil, our drug, this 5-FU, it ends up replacing itself into RNA, causing RNA damage. And then the RNA synthesis and everything can't carry out properly. So then our tumor cells aren't able to grow, which is great for the cancer patients. So there's other fluorouracil metabolites that incorporate into both RNA and DNA. So that incorporation into RNA really results in these major effects on both RNA processing and the function, sort of as was mentioned throughout the presentation. And here are the citations. I found that PubChem was really, really helpful. It's always been very helpful for a lot of case studies where we're talking about drugs. So I would encourage taking a look at that. Those were very helpful. And that's the end of my presentation.